Great. Hello. Thank you for coming to my talk. My name is Melissa Betters. I am a PhD student at Temple University in the biology department. And today I'm going to be talking to you about the environmental influence on shell morphology in deep sea gastropods. So when I say the deep sea, what I'm referring to is the part of the ocean below 200 meters depth. Despite this being the Earth's largest habitable ecosystem, it is one of the most chronically undersampled, both in regards to land as well as shallow water. And this is a real problem because the ocean today faces an unprecedented amount of anthropogenic threats, including overfishing, ocean acidification, and plastic pollution. And as we all know, being in this field of systematics, species classifications are foundational to proper conservation and academic efforts. Where I specifically do my research is along the Costa Rica margin, which you can see here. These sites are all of the sites that we've sampled in the past 10 years. However, my research focuses on just two of them, both Jaco Scar as well as Mound 12. What these sites are hydrocarbon seeps. So the Costa Rica margin is a tectonic margin where the Cocos plate is subducting underneath the Caribbean plate. And when this happens, chemical-laden pore water gets pushed out of that subducting plate and into the water column. The chemicals in this water can be used by microbes to undergo a process known as chemosynthesis, which is the primary production at these sites. And this primary production can support a whole host of endemic species that we're only now just discovering. One of these organisms supported by this primary production are gastropod mollusks. So as you can see in this image, they are highly abundant. They're also the primary grazers at these sites. Specifically to my research, I am going to be looking at the genus Provanna. Provanna are a very morphologically diverse genus. As you can see here, all of these organisms are from either Jaco Scar or Mound 12, and they're all included in this study. So during my first year of my PhD, I was tasked with classifying these organisms and determining which are distinct species. I got to asking whether these differing morphotypes are actually diagnostic of species, or if something else is influencing this morphology, and what that could be. So my question that this led me to ask is, to what extent does shell morphometry vary along environmental variables associated with both active hydrocarbon seepage at these sites, as well as depth? And my main aim of this research is to critically assess the reliability of shell morphology as a data source for taxonomy, and seeing what's phylogenetically informative and what's not. The reason I'm looking specifically at depth is because a lot of environmental variables have very predictable relationships with depth, including what you can see here, dissolved oxygen, temperature, as well as salinity. And this could be particularly relevant to my sites because Mound 12 occurs much shallower than Jaco Scar, about 800 meters shallower, enough to put them in distinct water masses. And of note is that Mound 12 occurs much closer to the oxygen minimum zone of the ocean than Jaco Scar. Furthermore, I'm looking at seepage because it's the main limiting factor of these environments. It's their food source, it supports the microbes on which these gastropods feed. So what you see here is essentially a map of Jaco Scar and Mound 12 of hydrocarbon seepage activity, denoted by ORP, or oxidative reductive potential, which we use as a proxy. So as you can see here, there are variations in ORP levels within sites. And what I'm interested in is whether these variable ORP levels can influence the morphology seen by these organisms. This data in particular is collected using the autonomous underwater vehicle known as Sentry. So how we collect biological data is a little bit different. We use the human-operated vehicle Alvin. And Alvin is equipped with a number of tools for biological sampling, which you can see here on the video on the left. It has an arm for grabbing organisms. It has pots which we can use to scoop up numerous organisms at once, as well as push cores and vacuum canisters for mobile organisms. And all of these are attached to the front of the sub. How I collect the morphometric data is by measuring the samples. So I've converted an image of one of my samples into this nice little illustration just for the simplicity of presenting. So I take a few measurements from each organism, the first being shell length, which is the longest point on the organism, shell width, the operculum opening, so this is measuring the shell, not the tissue, the operculum opening length, as well as the width. And I also chose to quantify the texture of the organisms because, again, there is a huge variation in the texture of these shells. So I measure the spikes of these organisms and average them to get a texture measurement. So I've got my explanatory variables being related to the environment and the response variables being related to the shell morphometry. The first thing I chose to look at was the effect of site. So looking at the difference between mound 12 and Hako scar. And what I found was pretty stark differences in both shell length, shell texture, as well as shell width. With mound 12 being smaller and less textured, and Hako scar being larger and much more textured. They tended to be more spiky and more bumpy. However, I wanted to get more down into the weeds and really figure out what was going on here and what variables were causing these differences. So I chose to do a multiple linear regression, both within sites as well as between sites. So between sites is encompassing all of the organisms that I have. So I chose my variables, tested my models, and the best fit model for shell texture specifically was accounted for with depth. Depth was the explanatory variable pulled out by the model as being the most significant. So this told me that shell texture did not seem to be correlated with hydrocarbon seepage, but these sites are 800 meters apart in depth. So I figured getting into within site analyses would give me a better picture of what was really going on here. So first, I did a multiple linear regression for Hako scar, the same process. This time, it pulled out temperature as actually the most significant explanatory variable. This was different than at mound 12, which pulled out oxygen as well as depth component again. I do think that the oxygen concentration variable is important here because of mound 12's proximity to the oxygen minimum zone, and that mound 12 is encountering much more limiting values of oxygen than Hako scar is. Then I ran a multiple linear regression again, this time looking at shell size, using shell length as the proxy. So again, I chose my variables, ran the model, and this time found that both depth as well as oxidative reductive potential, that proxy for hydrocarbon seepage, significantly contributed to the model. And this showed me that depth as well as seepage here seems to be important for shell size. I also ran a multiple linear regression again within each site. Within Hako scar, it pulled out temperature, oxygen concentration, and oxidative reductive potential as significant explanatory variables. Of note, all of these variables have to do with metabolism. At mound 12, it was a little bit a different story. The best fit model only included oxygen concentration. However, one thing of note here is that this shows an inverse relationship with oxygen than we observed at Hako scar. So where Hako scar shells increased with increasing oxygen concentration, at mound 12, shell length actually decreases with increasing oxygen concentration. And one reason why this may be the case is if you take a look at the x-axis, this shows that mound 12 indeed encounters much lower oxygen concentrations than Hako scar. Again, most likely due to its proximity to the oxygen minimum zone. So one reason this may be happening, why this inverse relationship could be occurring, is that these organisms are particularly well adapted to low oxygen conditions. However, I do want to study this more and really parse apart why this relationship is being exhibited the way that it is. So, in conclusion, what I've found in this study so far is that shell texture appears to be linked with depth. The deeper the organism, the more spiky or bumpy, the shallower, the smoother. And this is particularly being correlated with temperature, oxygen, as well as depth itself. And one thing that I think may be happening here is it could be indicating environmental filtering of species. And perhaps that texture is indeed phylogenetically informative. It is pretty common in the marine environment for species boundaries to occur very quickly across depth, as opposed to across horizontal distances. Shell size, on the other hand, appears to be linked to both depth as well as hydrocarbon seepage. And this may be related to food availability. Again, both of these things are correlated with metabolism. So the greater the ORP, the greater the bacterial densities on which to feed, the larger the shell. And this may imply that size ranges should not be used to characterize species at these hydrocarbon seeps, because it could be directly related to the availability of these hydrocarbons. So where I will plan to go with this research is to further investigate that inverse relationship with size and oxygen between the two study sites. I also plan on continuing my measurements. So I have only measured about 58 individuals so far, but I also plan to include more measurements themselves, including size to mass ratios, as well as radular morphology. I also plan on extracting DNA and sequencing barcoding genes to confirm genetic diversity and identity of these organisms. And with that, I want to thank you and give thanks to everybody who has helped me with this research, and as well as you for coming and attending this talk.